Welcome and thank you for joining us for the first AGRO Lunch and Learn webinar of 2023. This webinar series is possible through the generous support from Eurofins Agri-Sciences Services. So we thank Eurofins very much for their continued sponsorship. My name is Daniel Swale, and I'm an associate professor in the Emerging Pathogens Institute and the Department of Entomology and Nematology at the University of Florida, and I'll be serving as the moderator for today's webinar. Attendees should use the Q&A tool to submit their questions and comments, which will be answered at the end of the presentation. All right, so go to the next slide. I'll have a couple of slides to introduce for you prior to our speakers. So before I introduce our speaker, I'll highlight a couple of the benefits of becoming a member of the agrochemical division. AGRO plays an enormous benefit by enhancing your science, building your scientific network, and supporting your professional development. Please go to agrodiv.org and click on Become a Member to become an affiliate for $20 a year, which is regardless of your membership status with the ACS. All right, we have a pretty unique opportunity where this ACS Spring National Meeting is in Indianapolis this upcoming March, so this month, and it's a rare opportunity to experience agro technical content during the spring meeting. Registration is currently open, but the housing deadline is today. On Wednesday, the 29th, Corteva is presenting an agro division social event with refreshments, fun, networking, and prizes at their campus, which is just minutes away. If you have questions concerning the programming, please contact Ken Racky or Aaron Gross of Virginia Tech for agro programming questions. For the fall meeting, we have the standard excellent agro programming, as well as the rescheduled 50th birthday party of the agrochemical division. The abstract deadline is April 4th. If you have any questions about the programming, please again contact Aaron Gross of Virginia Tech, who is serving as the program chair for both the spring meeting and the fall meeting in Indianapolis and San Francisco, respectively. All right, so I'm proud now to introduce our speaker, Dr. Stephen Duke, who received his PhD in botany from Duke University in 1975. Since then, he has been mostly involved in weed-related research for the Agricultural Research Service, or ARS, of the United States Department of Agriculture. He was the director of the Southern Weed Science Laboratory of ARS from 1987 to 1996, and was the research leader of the Natural Products Utilization Research Unit in Oxford, Mississippi from 1996 to 2000. He retired from the USDA in 2020 and became a principal scientist at the National Center for Natural Products Research of the School of Pharmacy at the University of Mississippi. He is best known for his research on herbicide modes of action and natural products for weed management, such as his work on mechanism of action of PPO inhibiting herbicides, indithol, glyphosate, and other herbicide classes. He is also an authority on herbicide-resistant crops and allelopathy. He has over 500 peer-reviewed publications that have resulted in Web of Science and Google Scholar H indexes of 71 and 103, respectively. He has been the editor or co-editor of 11 books and co-author of a textbook on mode of action of herbicides, as well as a guest editor of several special issues of journals. He has been active in several scientific societies, having been president of the Weed Science Society of America, the International Allelopathy Society, and International Weed Science Society. He is a fellow of the WSSA and was awarded the Outstanding International Achievement Award of IWSS. He has also been the president and the chair of the Agrochemical Division for American Chemical Society, and has been the recipient of the highest award of IAS and is a fellow of the AGRO. He is also a fellow of ACS and the American Association for the Advancement of Science, and most recently, recipient of the Sterling B. Hendricks Memorial Lectureship. So without further ado, please help me in welcoming Stephen O. Duke for his presentation, Molecular Targets of Insecticides and Herbicides, Are There Useful Overlaps? Steve?
Okay. There we go. Thank you very much for the kind introduction, Daniel. And also thanks to AGRO for allowing me to speak today. As Daniel mentioned, my title is Molecular Targets of Insecticides and Herbicides. Are they useful overlaps? This is something that actually Daniel got me into about a year and a half ago. I don't seem to be able to change slides. Here we go. Here we go. Sorry. If you're interested in more details on this topic, this paper came out last month in Pesticide Biochemistry and Physiology. It has a bit more detail than I can get into this today. Also, there'll be a few things I mentioned in the presentation that are not in this paper, but I recommend reading the paper if you're interested in more detail. Note that Daniel is a co-author of this, as well as several other people. Many pesticides of one pest type have often come from pesticides developed from the chemistry of pesticides with another pest type. And the only review that I'm aware of on this topic is by Lamberth, published last year in Pest Management Science. An example of this is metallochlor, which is a herbicidal inhibitor of long-chain fatty acid synthase. It was modified slightly, and the R enantiomer of the modification had fungicidal activity. The S enantiomer still had strong herbicidal activity. And one of these enantiomers was eventually converted into metalaxyl, which is a fungicide, and a very important fungicide at that. So a herbicide with one mode of action was modified eventually to become a fungicide with a very different mode of action. There's some caveats to using this approach to finding new insecticides. Herbicides that are both good insecticides and herbicides may not be viable as insecticides because of crop damage. So in all cases of which I'm aware, insecticides that have come from herbicides have a different mode of action, except for one case that I'll discuss. Exceptions could be compounds that target non-crop arthropod pests, such as mosquitoes, sandflies, or termites. But the relative economic value of these is low compared to insects that are very important in agricultural production. Exceptions could also be when the target site of the plants and insects differ sufficiently to allow insect-specific and plant-specific molecules for the same enzymatic target. And also making the crop resistant to the compound could be possible with a pesticide that kills both weeds and insect pests. And if you could do this, you would have a compound that would reduce overall pesticide load because you'd be using one compound for both. This is HRAC's breakdown of the target sites of commercial insecticides. I've highlighted in green the ones that are in both plants and animals, and the ones in light green, acetyl-CoA carboxylase and mitochondrial uncouplers. There are actual compounds that inhibit both targets in insects and plants. The darker green are targets in insects that are not used as for herbicides. So I've noted the ones there that are actually used for commercial insecticides and herbicides. These mitochondrial targets, only uncouplers, are common to herbicides and insecticides. And these compounds are for the most part banned because of toxicity. They were fairly important pesticides back in the 50s and 60s. Y complex 1 through 4, mitochondrial complex 1 through 4 in plants, and ATP synthase have not been herbicide target sites. Is not known. If we look at the HRAC breakdown of target sites of commercial herbicides. The ones in green there are found only in plants. The ones in pink, I assume that's pink, I'm colorblind, on the right are the 10 target sites that are shared by both animals and plants. So these target sites are found in insects as well as humans, for that matter. Something that's not very often talked about. And several herbicides have unknown modes of action. And I mentioned the fact that the ACCase inhibitors and mitochondrial uncouplers are utilized by both commercial herbicides and insecticides. Here's a breakdown of the of 9 of those 10 overlaps, plus a few others, in terms of the genes in humans. HS there is the abbreviation, Drosophila melanogaster and Arabidopsis thaliani for the plant. And we have both the projected size of the protein, which are almost the same for each one of those in the plants and animals. And then the similarity with Arabidopsis being 100%, you can see it ranges quite a bit in terms of similarity. But a few of them are quite similar, especially alpha-tubulin. Note that there's about a 90% similarity in animals and plants. I mentioned the uncouplers. You see that there's a list of herbicidal uncouplers that are still theoretically used, at least in some part of the world. Dinoc is also listed by AGRO as an insecticide. All of the herbicidal uncouplers are dinitrophenols, whereas insecticidal uncouplers include some other compounds that are not used as herbicides. ACCase inhibitors belong to 3 chemical classes: the aryloxyphenoxypropionates, the FOPs, the cyclohexane diones, the DIMs, and then one compound that's a phenylpyrazolene. You see the chemical structures of those different chemical groups. The ACCase inhibitors of insecticides do not belong to those chemical groups. They are tetronic and tetramic acid derivatives. And interestingly enough, these compounds actually came from work with a herbicide. And this is sort of obscure literature, but the company that discovered these compounds was working with PPO herbicides that you see on the left, and they privatized those to some compounds that still had herbicidal activities, but were no longer PPO inhibitors. They were ACCase inhibitors, and they weren't such good herbicides, but they in fact ended up being the first of these ACCase inhibitors that I mentioned that are insecticides. And you can see a list of those that were developed from this, originally a PPO inhibitor. So we had a weak ACCase inhibitor that was never developed as a herbicide, from which the insecticidal ACCase inhibitors were derived. This shows that old chemistries can often yield new modes of action. This is an example. These are all diphenyl compounds. And you can see that PPO inhibitors, ACCase inhibitors, SPS inhibitors, plastid ATPase inhibitors, and enol reductase inhibitors all come from this chemical structure. It's interesting that triclosan, a microbial killer, is also quite toxic to plants and insects. So here we have a compound that's a diphenyl ether that is both herbicidal and insecticidal, but is not used as a commercial compound for either one of these uses. So ACCase inhibitor insecticides are available, but the herbicides also, the ACCase inhibitor herbicides are also active on insects. This is a paper from Israel showing that pheromone biosynthesis activating neuropeptides, their effect is inhibited by diclofop on the left there. And you can see on the right a dose response activity, inhibition of this activity with a triloxidem. And you can see that it's quite efficient at inhibiting production of pheromone biosynthesis. Another paper by the same group looked at a number of different herbicidal ACCase inhibitors and found very good activity there at the top, down to no activity for quizalofop. There was some concern that these herbicides might be affecting pollinators and butterflies. This paper showed that the effects were minimal on production of pheromones in 3 different species of butterflies. I'll next talk about glutamine synthetase. You can see there's a rather high similarity index in plants, insects, and humans for this enzyme. The only herbicide that's sold, that's a rather major one, is glufosinate, which you see there on the left, also called L-phosphenothricin, from which it was derived, a natural product. It inhibits glutamine synthetase in plants, which results in higher levels of reactive oxygen species like singlet oxygen, hydrogen peroxide, singlet oxygen production, and hydroxyl radical. And it works very quickly as any compound that causes rapid reduction of reactive oxygen species. It's also active on insects. This paper shows its effect on glutamine depletion in a caterpillar, and you can see that after on the left there, after treatment with glufosinate, you get eventual death and inhibition of a lot of processes. And the toxicity is not due to reactive oxygen species accumulation, because insects do not have photosynthesis or photorespiration. It's due to glutamine depletion. And here you see the visual effects of a very low concentration of this compound on a particular caterpillar. Other glutamine synthetase inhibitors have not been tested. At least we couldn't find any evidence of it in the literature. Bialofos is a small peptide that breaks down to L-phosphenothricin. It was sold in Japan as a herbicide, at least at one point in time. It might be active for the same reasons. Tabtoxin is a very active glutamine synthetase inhibitor. Methionine sulfoxamine is a synthetic GS inhibitor. And there are many other natural and synthesized compounds that effectively inhibit this enzyme. They could be even more active on insects than glufosinate. The only literature we've found of any of these compounds on the enzyme from an insect is mosquito GS activity is definitely inhibited by the synthetic methionine sulfoxamine. More work could be done on this as a target site since glufosinate is so active. A herbicide inspired by natural compounds, the ketones, which came from a compound pheromone produced by the bottlebrush plant. In ICI, back in the day, took this natural phytotoxin and developed the triketones, one of which is mesotrione, which is sold under a trade name that goes back to the genus name for the bottlebrush plant. All of the HPPD inhibitors inhibit hydroxyphenylpyruvate synthetase, dioxygenase rather, which produces homogentisic acid, which is a precursor for plastoquinone and alpha tocopherol. The phytotoxicities mainly due to loss of plastoquinone, which is needed for photosynthesis too. It's also a cofactor for phytoene desaturase, which is needed for carotenoid synthesis. And you can see the effect of the herbicide on a plant. It has no carotenoids, so you have a totally chlorotic, bleached looking plant, which is lethal. I should note that HPPD inhibitors are also used for pharmaceuticals at a very, very high cost. You can see the cost of a treatment of these midosome, which is a very close analog of mesotrione, inhibits human HPPD, and this prevents the symptoms of two hereditary diseases, one alkaptonuria and the other type 1 tyrosinemia. I won't get into the details of those diseases, but HPPD inhibitors do inhibit the effects and are quite effective. This shows that these compounds are active on the human form of the enzyme. If you inhibit the enzyme in insects, you can prevent the toxicity, or you can cause the toxicity of tyrosine in blood-fed insects, mosquitoes, ticks, bedbugs, sandflies, etc. And this high level of tyrosine accumulation when you inhibit the enzyme causes toxicity and death. And this has been shown in a number of papers by Ramirez et al. That are all outlined in the paper that I mentioned. Very effective on blood-fed insects, but not on insects that have not fed on blood. Serine threonine protein phosphatases, you can see there are high levels of similarity between that enzyme in plants and insects in particular, not as much as in humans. The only herbicide that inhibits this enzyme may have come from cantharidin, which is a natural product produced by an insect. It breaks down the cantharidic acid to inhibit the enzyme, and you can see that's a very, very close analog of indithol, which is a very old herbicide, only differing by 2 methyl groups. And we published a paper a few years ago showing unequivocally that that's the serine threonine protein phosphatases are unequivocally the target site for indithol in plants. And in fact, indithol and cantharidin inhibit the enzyme in plants very effectively. You can see in both Arabidopsis and duckweed. Interestingly, indithol's also quite active against insects in the one paper that we found where people had tried to see if it was effective, and you can see that it is fairly effective. No work since this paper in 1987 has been done on this, as far as we could find. In the same paper, the authors found that a inhibitor of very long chain fatty acid synthesis in plants, thiobencarb, inhibits cuticular hydrocarbon deposition in an insect, and was also quite active as a herbicide. PPO inhibitors is a rather large class of compounds that are used as commercial herbicides. Several years ago, there was a lot of interest in compounds that might cause porphyrin accumulation in both plants and animals. This ACCase published book that we edited covered a lot of this literature that was on the topic, but the only compound that's actually causing accumulation of porphyrins in pests are PPO inhibitors. They have a very unique molecular target site. After the entry of the herbicide into the plant, it inhibits protoporphyrinogen oxidase, which is a plastid or chloroplast enzyme, which causes the substrate to leave the plastid. Substrate is protoporphyrinogen 9. It's converted to the protoporphyrin 9, which is a normal product of the enzyme in the chloroplast, but it's produced outside the chloroplast, where in the presence of light and molecular oxygen, it produces singlet oxygen and a cascade of other reactive oxygen species, causing rapid degradations of lipids and membranes. So you've got an enzyme causing accumulation of the substrate rather than the product. And it's a very, very effective herbicide. Some of the best PPO inhibitors are active at only a very few grams per hectare. You can see there is some similarity in the enzyme between plants and animals. Early work on this PPO inhibitors found that it was quite active on human PPO, even though the similarity is not that great. But we have not found any publications on the activity on insect PPO. As a matter of fact, there's virtually there are no publications on PPO insects, especially on PPO inhibitors. Synmethylin is an interesting herbicide, probably coming from one for cineole. It was discovered by Shell many years ago, and it's through several iterations has ended up with BASF. It's been sold under a number of trade names. It's sold as Luximo now. And recently, BASF published a paper showing that it inhibits acyl ACP thioesterase or fatty acid thioesterase in the plastid, which inhibits the synthesis of mid-chain length lipids in plants. BASF also recently published a paper showing that a number of herbicides for which we had no known mode of action also inhibit the same enzyme. And you can see that there's not an awful lot of similarity between the enzyme in plants and animals, but there's no evolved resistance to herbicides that act at this mode of action. The only thing we found regarding this enzyme in insects that indicate that it might be a decent target site for inhibitors of that enzyme is that RNAi knockout of the gene that's expressed in the integument of an insect is lethal to it. Tubulin is a target site for a number of older herbicides, and there are reports of mild toxicity of alpha tubulin binding herbicides being toxic to insects, but not terribly toxic. Podophyllitoxin, which is not an insect, and it has not been used as a precursor for an insect, but it has been used as a scaffold for insecticide design. We found in our lab that it is a tubulin inhibitor as a phytotoxin, and a fairly good phytotoxin. And as I said, it's been explored as a template for insecticide. You can see the homology there in alpha tubulin is quite high. So it theoretically could be a insecticide target site. There are compounds that are termed by HRAC as inhibiting microtubule organization. These include barban and chlorprofam. Chlorprofam causes complete mortality in a parasitoid wasp, and chlorprofam is effective against the potato tuber moth. So, and it's barban's reported to be an insect chitin synthase inhibitor, and chitin synthase has been reported to be involved also interacting with microtubules, so indicating that there could be some overlap there with insecticides. That concludes the 10 plant target sites for herbicides that there could be overlap with insecticides. There are a lot of novel target sites especially among natural products that could be used for both insecticides and insecticides. We listed a number of those in a publication almost 10 years ago. Here you see a list of them. One of them is actinonin, which interferes with protein production in organelles, both mitochondria and chloroplasts. It inhibits peptide deformylase, and it's quite active here, as you can see on plants. Actinonin has been patented as a herbicide. You can see it's fairly active also in growth of the Australian sheep blowfly larva. Here's one that's not mentioned in the papers because people say it's just too ridiculous to even consider these compounds as pesticides because they're so toxic to humans. But there are a number of natural compounds, fumonisins, L-toxin, australofungin, and others, which inhibit ceramide synthetase, which could loss of ceramide can cause membrane dysfunction and apoptosis, but we have good data that show that it causes very high accumulation of precursors of ceramide synthase, and that these very quickly cause membrane dysfunction and also apoptosis. This is from one of our earlier papers showing that in a plant, you see there the fumonisin and L-toxin cause very rapid accumulation of ceramide sphingoid bases, which are very phytotoxic. Here you see the effects of fumonisin on yellow mealworm larvae. It's quite phytotoxic at very low concentrations to them also. We did quite a bit of work trying to find analogs that would be not toxic to humans, but toxic to plants. We could improve things about tenfold, but that's really not nearly good enough. It is still possible that with modern methodologies, the work we did was almost 30 years ago. With modern methodologies, you might be able to improve that ratio to make something that's not toxic to humans. Hydanocidin is a compound produced by Senkyo, a pharmaceutical company in Japan. There's quite a bit of interest because they showed that it had good activity on 7-monocot and 10 dicot species. Work outside of Sancho showed that it works as a herbicide through inhibition of adenylosuccinate synthase, and that it does this by being phosphorylated has to be phosphorylated to be active as an inhibitor. So it's a pro-herbicide. It's also quite active on mosquito larvae. It could be either a herbicide or an insecticide. Several companies spent quite a bit of effort producing analogs, trying to find something that could be a commercial herbicide, but it never got developed. A compound that we've worked on, or one compound, is from a bioherbicide produced by Marone, or discovered by Marone Bioinnovations. One of the compounds in their microbial product is romidepsin. Here you can see a very complex molecule, and it inhibits histone deacetylase, which in insects has quite a bit of similarity with the molecule with a target site in plants, 73%, not as much as in humans. We found that it's a very good inhibitor of Arabidopsis with a very low I50, and it's very active inhibitor of histone deacetylase in Arabidopsis. It also is very active against blowfly. You can see there that it's active at a very low I50 there also. So that's a compound that could be active on both. There are pharmaceuticals that are active on both plants and insects, and I mentioned the HPPD inhibitors as having overlapped with pharmaceuticals, mesotrione and nitinosone being very, very similar molecules. Inhibitors of 3-hydroxy-3-methylglutaril CoA reductase, or HMG reductase, are very well known statins to reduce cholesterol in humans. They're also phytotoxic, indicating this could be a novel mode of action. They inhibit production of stigmasterol and beta-sitosterol in plants, both of which are needed for proper plasma membrane function in plants. You can see that there's a fair level of similarity of this enzyme in plants, insects, and animals. Here you can see that here are some of the pharmaceutical HMG reductase molecules that are used as pharmaceuticals. These compounds inhibit the non-mevalonic acid pathway, which is required for juvenile hormone biosynthesis in insects, and these two sterols involved in plasma membrane function in plants. Here you can see in a paper published a few years ago that two of these statins, atorvastatin and lovastatin, inhibit both the plant sterols in duckweed and are quite good growth inhibitors. The sterol synthesis coming a little before the growth inhibition. This is some work from Josh Mills' lab in Australia, very recent work, showing a lot of analogs that they produced having very, very active growth inhibition against Arabidopsis, and they're trying to refine these compounds to be even more active as herbicides. Compactin is a very good HMG CoA reductase inhibitor of juvenile hormone synthesis and inhibiting production in hornworm by 50% at 10 nanomolar. This is compactin. And Zhang et al. Published fairly recently a paper showing fluvastatin analogs and their effect on larval mortality of insects. And they found very good activity against juvenile hormone biosynthesis. So herbicides, some natural products, and some pharmaceuticals can all have decent herbicidal and insecticidal activity, and the molecules be manipulated to have improved herbicidal and or insecticidal activity. So there are many more herbicidal molecular targets held in common with insects than vice versa. There are some fairly insecticidal and herbicidal natural and pharmaceutical compounds that act by targeting the same enzyme. These compounds have generally not been optimized for insecticide or herbicidal activity, at least not in the published literature. Opportunities exist for discovery of insecticides from herbicidal compounds that share molecular target sites with insects. And some herbicides are no doubt providing some additional pest management benefits by their effects on insect pests in the field, something that really hasn't been documented, but it's almost certainly occurring, especially with ACCase inhibitors and perhaps glufosinate. And with that, I thank the co-authors to the paper and the many people that were involved in some of the data that I showed that came from our publications. And thanks for listening, and I'm open to a few questions now.
Great. Thank you very much, Steve. If you'll maybe stop sharing your screen.
Let's see, how do I? Oh, shit. Okay.
All right. So thank you very much for your participation in the webinar. First and foremost, I'd like to thank Stephen Duke, our speaker, for the incredible talk he just gave us here at Fenn's AgriScience Services, our webinar coordinator, Kyle Sawyer, and the webinar committee for the work on this program. Let's see if I can get this pulled up. All right. So you may be asked to complete a follow-up survey after the webinar, and we look forward to your feedback. Future webinar planning is going on right now, so please contact Salito Sumilong if you're interested in supporting our efforts to publicize this series. And with that, I would be happy to, well, we can take any questions. Please type them in the Q&A box on Zoom, and we can get to answering those. Let's see, I'm trying to. All right, as we wait for those to come in. All right, Steve, I'll ask a question for you. How much, or can you speculate on the overlap that there is between herbicide modes of actions and potential fungicide targets?
That's something that I've thought about getting into. Certainly, there is quite a bit of overlap. For example, glyphosate, the molecular target of glyphosate is found in all fungi, and in most cases it's quite sensitive to it, especially rust. In fact, at one point, I think Monsanto, they published some papers showing it was quite effective on rust diseases in Roundup Ready crops, and they considered putting it on the label as a fungicide against rust diseases, for instance. Asian soybean rust in Roundup Ready soybeans. And there are quite a few other target sites that are found. I may get around to writing a similar paper along the same lines for fungicides in the next year or so.
All right, so starting to come down some of the list here. One of them, Steve, does cross-reactivity of herbicides as insecticides endanger any registrations?
It could. Yeah, that's sort of a new concept. As I mentioned, there was concern when it was shown that some ACCase inhibitors were quite active against some insects, and there was concern that they might affect butterflies and or pollinators. So you'd have to go through the same registration procedures for a compound that was active on herbicides and insecticides. So the dossier for approval would be even more massive than what's required now for just one type of activity. So that's there would be concern, yeah.
Excellent. So we have a couple of questions regarding the protein sequences. So we'll start by regarding the table showing the comparison of protein sequences across the three species. Is it based on full-length proteins or based on the active sites, binding sites? And on that topic, are there any better predictive technologies that might enable prediction of cross toxicity as a predictive measure for cross toxicity?
Very good question. Those similarities are based on full-length proteins, not on the binding sites. We didn't get deep into the weeds on binding site sequences, and if we look at those, I expect where we do see good activity for both insecticidal and herbicidal activity, we might see even better similarities than the full length. But we had a rather long paper at the point we got to the point that we did, and that could have increased the length of the paper quite a bit. So we didn't get into that. But I expect that if a company wanted to pursue this, that would be the next thing to do.
All right, so we'll take a couple more questions here. What is the advantage of using some of the known herbicides as insecticides, given that they may increase or continue the selection pressure of weed species?
Yeah, if you had I don't think any of the known herbicides are going to be used commercially as insecticides. If you developed a compound that based on one of these on a common target site, this would add to the selection pressure, at least in the weed sector, further selection pressure for target site resistance for that particular target site. So that would be that would be a negative, unless you found a binding site on that particular enzyme that is not overlapped with the currently used herbicidal inhibitors for that target site. Yeah, that's a concern for sure.
Right.
One might say the HMG CoA reductase inhibitors would add a new target site, so something like that would be both insecticidal and herbicidal. You're starting fresh. There is no resistance that we know of to inhibitors of that particular target, so that might be a better approach. But resistance management, if we did have something that valuable, resistance management would be a very big part of the introduction of a product like that.
And we have a question from Marja. Based on your experience, is there a safety margin or at least a minimum difference in IC50 values to safely use an herbicide as an insecticide?
It's going to vary dramatically depending on the compound in the particular species. Something that we know virtually nothing about for any of the examples that I gave.
All right, it seems like questions have started to taper off, so we'll give it one more minute to see if there's anything that rolls in. If not, in the meantime, I'll introduce our next upcoming webinar at 12 p.m. on April 12th. We're going to hear about the use of digital images in field experiments. So please be sure to join us with that. And on the following one after that will be on May 17th, talking about recruiting talent in the agrochemical business, particularly with focus on Corteva. All right, it looks like we have no more questions coming in. So please, again, join me in thanking Steve Duke for the excellent presentation. And thank you for everybody behind the scenes on the webinar team. Thank you very much.
Thank you.